We did a paper on inhibitors of fatty acid amide hydrolase. Okay, so part of the system for the fatty acid amide hydrolase are cannabinoids and endocannabinoids. So cannabinoids can be any one of approximately 113 compounds that are derived from cannabis, and then there are endocannabinoids, and they are endogenous ligands for cannabinoid receptors CB1 and CB2, meaning that they're found naturally, they occur naturally in our body systems. And CB1 receptors can be found in the brain and in the body, and CB2 receptors are usually found in the immune system and the gastrointestinal system, and they can be synthesized from fatty acids. And CB1 and CB2 can be used to treat inflammatory and neuropathic pain. And in the paper, they had mentioned that one of the agonists of the CB1 receptor called 2-arcodiol glycerol are considered to be endogenous cannabinoids, and they've shown to regulate several neurobehavioral processes like pain, motility, cognition, and feeding. And anandamide takes on the role of an endogenous ligand for the central cannabinoid, the CB1 receptor, and that recognizes 9-tetrahydrocannabinol, which is THC, the active component in marijuana. They are also incredibly lipophilic, meaning they do not travel far like hormones do because they don't like water, and they can be found at intersections between organ systems to allow communication.
So in the endocannabinoid system, there are 3 components: the endocannabinoids, receptors, and the enzymes. So kind of highlighting what Naya said, they can help reduce inflammation, and endocannabinoids are broken down after they do their job. So more on the CB1 and CB2 pathways, or the pathways. The CB1 is found mainly in nerve cells, brain, and spinal cord, and it's present in the hypothalamus, and when it's present there, it affects the appetite, and when it's found in the hippocampus, it influences memory and processing of the brain. For the CB2 pathway, it's found mainly in peripheral nervous system, and the role of it is to keep the body's immune system healthy and well-functioning by regulating the release of cell signaling molecules, and these molecules boost the communications between cells and accelerate the movement towards the area that's inflamed that it's trying to reduce the inflammation of. So these are just linked to a variety of processes, and all of these processes relate to maintaining homeostasis somehow.
So this is the signaling pathway of CB1. So like they said, CB1 is highly expressed in the central nervous system, meaning the brain and the spinal cord, and CB2 is more in the peripheral nervous system. So they operate through a process known as retrograde signaling, which means they travel, these receptors the cannabinoids travel from the postsynaptic neuron back to the presynaptic neuron. So that's backwards from what most neurotransmitters do in order to pass down a signal. So during periods of like intense neural activity, such as feelings of anxiety or fear or a pain response, calcium 2+ collects in this postsynaptic neuron, and this increase in concentration causes the synthesis and release of endocannabinoids like AG2 and AEA from lipids. So this buildup in calcium in the postsynaptic neuron essentially triggers these cannabinoids to be released. And then they go back to the presynaptic neuron, so traveling upwards, and bind to either CB1 or CB2, depending on the location of the response. And they cause a variety of these downstream effects. So a lot of them are actually inhibitions. So they can inhibit adenylyl cyclase, which is that AC right there, which converts ATP to a secondary messenger called CAMP, which in turn can turn on protein kinase A. And these are involved in a variety of functions related to cell survival and death. So this pathway is actually inhibited by the presence of these cannabinoids. They inhibit the influx of calcium into the cells, notated by this voltage-gated calcium channel, which essentially prevents more of the pain response from being transmitted downstream. Because of that buildup in calcium cells, it's because of that voltage that allows these signals to be passed through neurons. So since that can no longer be passed through, since the voltage is no longer reached, because of the lack of calcium ions, pain signals and anxiety and fear responses are no longer transmitted throughout the brain, which is why these work as anti-anxiety or kind of pain relieving. And they all in all just modulate the expression of neurotransmitters and interact with these basic cellular functions. So like PI3K, which are a family of enzymes involved in cell growth and proliferation, etc., and are also an interesting kind of cancer target as well. So this pathway is involved in a lot of just cellular function, but mostly for this case is involved in kind of tampering the response with pain.
And so basically what they found is they wanted to explore how THC can be used to relieve pain and anxiety in cancer patients, and but they wanted to do that without the psychoactive effect of the THC. So what they did is they found this AM11542, which is a THC analog, and you can see that the inhibitor binds in the same way, or the analog binds in the same way as THC, and it's right in the pink right here. The yellow is cholesterol, and the cyan way in the back left is the flavin mononucleotide. And you can just see that it's in the center of the enzyme, showing that endocannabinoids don't bind on the surface. And the crystal structure just reveals important binding patterns that the researchers use to figure out potential inhibitors. So I did a little more research, and Naya, do you want to go to the next slide?
Oh, sorry.
And so I found these like cool figures, and then like you can kind of see further in like the one furthest to the left, you can see how the analog adopts the L-shaped conformation is like, and is really inside of the enzyme, like it's like very deep into the enzyme rather than on the surface. And then the one in the middle, it sums up all the interactions between AM11542 and the enzyme. So you can see like the
receptor.
The receptor, sorry.
Yeah.
You can see like the purple dots are the positively charged interactions. The cyan is the polar interaction, and green is the hydrophobic interaction. And the purple arrow right here shows H bond, and then the green line shows pi-pi stacking. And then the next figure shows more interactions just with more labeled parts of the receptor. So most of the interactions are hydrophobic, as kind of seen by all of these green dots. And they're also aromatic, and they consist of mostly of the residues ECL2, which is extracellular loop 2, and then helices 3, 5, 6, and 7.
Nice.
So what is fatty acid hamid hydrolase? It's an integral membrane enzyme responsible for the principal degradation of endocannabinoids, including anandamide, or otherwise known as AEA, which is the endogenous cannabinoid that binds in the same place as THC in the brain. And it's a member of the serine hydrolase family of enzymes. So basically, it's an integral membrane hydrolase with a single N-terminal transmembrane domain, and it destroys anandamide. So researchers wanted to develop an inhibitor that would block this destruction of anandamide, which would allow for elevated and sustained levels of AEA, which would help reduce pain in cancer patients and reduce anxiety.
So how FAAH works is just like its name implies, fatty acid amide hydrolase. It's going to perform hydrolysis. So it's going to like throw water at a molecule, and that molecule is going to split. So mainly it does this to fatty acid amides. It performs hydrolysis on those. And so yeah, it's a known as a catabolic enzyme because of this. And yeah, it just breaks down the fatty acid, and in our case, when it does that to AEA, it's not good, because we want those AEA levels to, like Lindsay said, like maintain a good kind of mental health care of ourselves. And on the next slide, we have the acylation step of the FAAH mechanism. So this triad of the serine, lysine, serine were all used together to perform the acylation step here. So the first serine, the serine 241, that's the one kind of at the top there, is going to be the nucleophile that's going to move electrons from its oxygen to the carbonyl. The lysine is going to play a role as both an acid and a base in this mechanism, helping to remove and add hydrogens where it's needed. And then the serine 217, so when that's mutated to alanine, it's going to produce some FAH mutant to reduce the hydrolytic activity and the FP reactivity. So this reduced FP reactivity is going to be indicative of a weakened nucleophile, which is why that serine wasn't the nucleophile, the serine 244, the serine 241 was. So altogether, this triad just works very well together to properly understand of this molecule.
Nice.
So the curious case of FAH out. So there was a woman, her name was Jo Cameron. She's 71 years old, and she had, at that point, had not experienced really any pain or anxiety during her life. Even during childbirth, she said she felt pressure, but it kind of felt like a tickle. And doctors had kind of looked into her case once she had a surgery on her wrist, and she didn't need any painkillers and wasn't in any sort of pain. And what they found was that there was a microdeletion in an FAAH pseudogene called FAAH out. And so we all have FAAH out, but what happened on Jo Cameron's was there was a deletion on one of the genes. And so then what doctors sort of start to realize with that is that it could have an epigenetic role in regulating FAAH transcription. So I guess if scientists can figure out what can medicinally inhibit that part of the gene, it could be used as a drug target. And because less FAH expressed causes an increase in concentration of anandamide or AEA, which can lead to reduced pain, sensation, and lack of anxiety and fear, which is what Jo Cameron was experiencing. And that kind of leads us into FAH as a drug target. So like I said, any genetic or pharmacological inhibition of FAH in general can produce effects that can help with pain relief, anti-inflammation, reduce anxiety or depression. And it doesn't, without, I guess, with the side effects of direct cannabinoid receptor agonists, like drugs that are used in anti-nausea for chemotherapy. And it doesn't target opioid receptors, but it can still reduce pain and anxiety. And that's pretty great because opiates can be very addictive. And they can inhibit the degradation of endocannabinoids rather than introducing outside cannabinoids that could produce adverse effects, meaning that instead of adding, elements of THC, which could lead to slow motor function or drowsiness, things like that.
They found a bunch of potential inhibitors, and one of the ones that they found that were the most effective was compound 10, or otherwise known as PF3845. And what they found was that it covalently binds to FAAH, and it leads to a non-reversible type of inhibition. And some key structures that led to the inhibited potency is the trifluoromethyl substituent on the biaryl, which is in red, and then the biaryl is in pink, and then the 3-aminopyridine leaving group in blue, and the carbonyl in green, which acts as the electrophile in green when serine attacks. Nice. And so what they found was that the piperidine portion in black occupies the acyl chain binding pocket of the enzyme, and the covalent bond occurs between the carbonyl and serine 241. So in the mechanism, on the next slide, what happens is the serine acts as the nucleophile and attacks the carbonyl, and the electrons go to form a carbanion, and then they reform the double bond, and the nitrogen group is kicked off as a leaving group, and the carbanion linkage forms between serine 241 in the compound 10, which is seen in blue.
So like Lindsay just said, in explaining the mechanism, this is how compound 10 or PF3845 actually binds to FAAH. So in the structure on your right, you will see the full structure of inhibited FAAH with the surface kind of transparent, so you can actually see some of the internal mechanics of this enzyme. So in yellow, way in the back, that is lysine 142, which, like Emily mentioned, plays a key role in the hydrolysis of AEA. In blue are serines 241 and 217, and that hot pink dot is just a sodium ion that is complex to the enzyme. So if you look at the structure to your left, that's a more zoomed in picture of how the compound actually binds. So it's bound, as you can see, to serine 241 in that carbamate linkage. There's that piperidine ring that's kind of shaped like a chair. And then there are also multiple other non-covalent interactions that lead to an increase in potency because of it. So there's a strong aromatic CH-pi interaction between phenylalanine 192 and the aromatic ring. So phenylalanine does have an aromatic ring on it, so that is why it's able to kind of attack in like that diagonal position to allow those hydrogens to kind of form an interaction with the pi bonds inside the aromatic ring of the compound. And then there is a 20-fold improvement in potency because of just van der Waals interactions of compound 10, specifically that trifluoromethyl portion and the hydrophobic chains. So because of it's able to kind of angle downwards and occupy more of that space within the pocket, it allows for more increased contact within the binding pocket, which increases potency, because the more contact you can have and occupy that particular binding pocket where AEA normally is, then the more potency you can have in inhibiting this enzyme. So now the more interesting part is actually how they created PF3845. It is a 4-step process. So step 1 is nucleophilic aromatic substitution, starting with 3 bromophenol and 5-trifluoromethylpyridine, seen on the bottom. So they stirred this overnight in DMF and K-carb at 110 degrees Celsius to yield this final product, where the chlorine was substituted and the oxygen was bound in that position to make the ether linkage. And this product is called 2,3-bromophenoxy 5-trifluoromethylpyridine, and it had a 95% yield as an orange oil. And this compound was carried on into the next step. So step 2 was actually a 3-part process. The first was the reduction of this aldehyde on the starting compound down to an alcohol using what's known as 9-BBN, which is 9-borobicyclo-3,3,1-nonane, shown in the box off to the right. And then they used sodium hydroxide to reduce that alcohol down to an alkene. It's like typical of a 1 reaction. And third, they did a Heck reaction. So if you remember from the first compound, had that bromine substituent on the benzene ring, that was what was used to be coupled with this newly formed alkene on this added product. So they reacted it under nitrogen for 16 hours to form this bond right here, that alkene linkage, and they purified it via silica gel column to yield a 75% yield of a clear oil. And as you can see, that's the really long name of their new compound. So step 3 was just a deprotection, the removal of this Bach group on the piperidine nitrogen, but somehow the alkene disappears and we don't know how. So that was interesting, but they did this under dissolving the first compound in methanol and DCM and then adding HCl dropwise to remove this Bach group just because of the addition of acid, and then redissolved it in toluene, and now you have this hydrochloride kind of bound to the outside. And this was a fluffy white powder, so it was vacuum filtered, and they didn't give a % yield, but 2.28 grams of this product was carried on to step 4. And then step 4, this final step, was the addition of an isocyanate to form a urea and the neutralization of the HCl. So it originally yielded an oil, but it was placed under a high vacuum to form a foam. The foam was dissolved in hot diisopropyl ether and then dried in a 40 degrees Celsius oven to finally give a white powder. So this is just a simple reaction with dichloromethane and diisopropyl ethylamine, and here's that isocyanate group they added. and they quenched it with sodium bicarbonate to remove that hydrochloride group. So on the next slide, you'll see the mechanism as to how this actually forms. So the lone pair on the nitrogen of this ring attacks the central carbon in the isocyanate, pushing the electrons onto that nitrogen to give a negative charge. Then DIPEA, so diisopropyl ethylamine, comes in and uses its lone pair to deprotonate the positively charged nitrogen originally in the ring, and subsequently reprotonate the negatively charged nitrogen to form that linkage. And that is how the urea is formed, and this final compound is given with an 82% yield. So that's not their total yield, but that is the yield from step 4. However, the percent yields from the majority of this reaction were over 70%, which is fantastic. And another thing to mention about this particular synthesis is that there is no stereochemical concerns. So they weren't trying to actively seek out one particular enantiomer or, in previous cases, a tripisomer. So this actually made the synthesis a lot more hassle-free.
And so how they measured the inhibition of compound 10 was they used an assay using oleamide instead of anandamide, which, although the initial experiment used anandamide, the use of oleamide is still valid. They actually chose to use oleamide instead of anandamide because urea is a side product, and the hydrolysis of oleamide, which is later used as a substrate in the enzymatic assay. So when FAAH catalyzes the hydrolysis of oleamide to oleic acid, NH4 is a side product, and then that NH4 is used in the reaction involving alpha-ketoglutarate and the enzyme GDH, or glutamate dehydrogenase, into glutamate. And in this reaction, NADH is oxidized to NAD+, and that reaction is what is monitored. And so they monitor the reaction at 340 nanometers to measure the amount of NADH in the reaction flask, as it is proportional to the amount of oleamide hydrolyzed to oleic acid. So the higher the absorbance is, it means that the inhibitor is more potent because less NADH is being converted to NAD+, because there's less urea being produced as a side product. And so what they did is they did 2 reactions, and they threw all of these materials into 2 different flasks, and one flask had the compound 10, or PF3845, and then they monitored the reaction at 340 nanometers for 40 minutes, and they found that the graph of their absorbance exhibited a curvature similar with an irreversible mechanism of inhibition.
Okay, so inhibitor and potency. So there are 2 parts to the sequence that make covalent enzyme inhibitors successful. So the first part of the sequence is reversible, and that's where an inhibitor will bind non-covalently to the target protein or the target enzyme. And it's usually where on the inhibitor, the electrophile is close to the nucleophile to a specific nucleophile on the target protein or enzyme. And the strength of the affinity bond can be measured using the Ki constant, which is also called the equilibrium dissociation constant. And it can indicate how potent an inhibitor is. And then in the second part of the sequence, that step is irreversible, and that's where the inhibitor forms a covalent bond to the either target enzyme or target protein. And this can be measured using the Kinact, which is the rate of enzyme inactivation. And so it's basically it's the rate constant for the conversion of the initial complex to the final complex, the final covalent complex. And so basically it's just measuring how quickly or slowly the conversion can occur. And so then when we're looking at the values and what they mean, Ki values that are smaller tend to indicate that binding is tighter. So that means that the inhibitor is more potent. And then for Kinact, if the covalent bond forms faster, then the number will be bigger, and if it forms more slowly, then the number will be smaller. And then as for Kinact, or Ki over K inact, the number can be big if the Ki is small, and it can also be big if the molecule that form the covalent bond happen quickly. And you can see in the compound 10 that this number is pretty big. So either one, I guess one of the two of these numbers had to be either it had to have either happened very quickly, or the potency was very high.
Nice.
Okay, so they used a proteomic technology called activity-based proteomic profiling. And so essentially what they did was they used a chemical probe, and in this case they used a fluorescent probe, which they use FP rhodamide. And this fluorescent probe has the ability to bind to any sort of serine hydrolase and display the interaction between compounds and target proteins or enzymes. So basically the three parts of the AAPB are, or the ABPP are a reactive group, a linker site, and a reporter group. And so essentially what they used, how they did it in this experiment, was they wanted to look at whether or not the small molecule inhibitor, PF3845, would selectively bind to just the FAAH. And that takes us into our first assay. Okay. So basically what they did here was they took mice that were pre-treated with inhibitors, PF3845 and a urea inhibitor that's very similar to and pretty selective to FAAH. And so then these mice brains, I guess, were labeled with the FP rhodamide. And I'm just going to abbreviate and say FP because that's just,
sure.
Okay. And, but I want to kind of focus on this first figure here. So, so basically what happens, so if they use various serine hydrolases, as I mentioned before, that FP has the ability to bind to any serine hydrolase, and there are tons of serine hydrolases, FAH being one of them. And so basically if the serine hydrolases, if the FP binds to one of the serine hydrolases, it will show up with a band on this gel. And if it doesn't, then no bands will be present. So you can see here in the fatty acid lane that after, almost 24 hours, that no FP had bound to it, but on the other serine hydrolases, you can see all these dark striations, meaning that the FP did bind to these serine hydrolases, meaning that PF3845 is selective for FAH because it wasn't able to bind to any of these other serine hydrolases. And you can also kind of see similar data. PF3845 is a little bit better than the urea compound because the line kind of showed up earlier, later than, this one. Okay, so then in this second assay, so they didn't necessarily use activity-based profiling, they used more so click chemistry. And so initially what they did was they converted the PF3845 into its, is alkanated the right word to use, to say what these would, what it would be?
Sure. Like the alkyne version, I guess, or
Okay.
something like that. Yeah, sure.
They convert it to the alkyne version, and similar to the urea compound. And so what they wanted to do was demonstrate again the selectivity of an inhibitor. And so then they convert it to the alkyne form. And basically what they did was they took mice that were injected with either regular FAH or FAH where the gene that expressed that was completely deleted, so there's no FAH present. And then what they did was they used click chemistry to attach on the FP probe, and any protein that had that attached on there would show some sort of molecular band. And so from the end of this experiment, they kind of realized that the alkyne version of the previous compounds were selective because there was no striation where FAAH wasn't present.
Nice.
Okay, and in this third and final assay, they basically tested like the levels of AEA or other fatty acid amides in the presence of the PF and the UR. So these 4 graphs at the top represent, like from left to right, the presence, the what's left over of AEA, PEA, OEA, and another FAAH inhibitor. But the most important for us right now is the first graph that represents how much AEA was present. So basically, the higher the graph is better because that's going to say that there's more AEA. And so just to kind of like sum this all up, FAAH degrades AEA, and that's bad. You don't want that. So the goal of the researchers here was to find or create some sort of inhibitor that would prevent FAAH from doing that. So in this case, it says in this graph, the PF3845 was higher. So that inhibitor was better at blocking the job of FAAH so that more AEA could be produced. So this was good, and it was just much better, more effective at blocking it than the UR compound was. And as we know, this sad story of how they figured this out was they had to take the mice that were tested and they cut off their heads and they basically just tested their brains literally to see like the amount of AEA that was left, and compare it between those that were tested with the PF and those that were tested with the UR inhibitor. And then lastly, so is PF3845 an effective painkiller? So it was found that 1 milligram per kilogram of the PF3845 was found to produce almost a complete blockage of the FAAH activity, which is wonderful. And it had almost maximal elevations of AEA, but the efficacy was not observed at this small of a dose. And that's just because the kinetic activity of FAAH at this small dosage couldn't really be noticed. So you can't really tell how effective it was in that case. So entelodynia is a symptom that you feel, or a symptom where you feel pain from something that doesn't normally cause pain. So again, what they did in this assay is they induced this random pain using the PF3845, and the antagonists for the central CB1 and the peripheral CB2 receptors were able to contribute to a significant reduction in the entelodynia activity of the PF3845. So overall, PF3845 was able to block inflammatory pain responses in the CFA model, which is just this mice pain-induced model. And they were able to do this using the cannabinoid receptor-dependent mechanism that involved both the CB1 and CB2 receptors, like those that are found in the FAAH inhibitors that we saw. And currently, this drug is titled as an investigational new drug, and it's being investigated to be used possibly in humans as a potential drug for pain relief.
Oh,
Questions?
nice. That was so much fun.